something. All right. Yeah. All right. Okay. We are writing. Someone is saying that wrote codominancy. The reason why they might not give you codominancy is because they say the intermediate. And there on our distinction material, let me just clarify for you, so that because also this one could be a disturbing factor, that people could mix it with the codominancy and the incomplete. Okay, supposed to be at the beginning here of yes, at the beginning here. Okay, yeah, supposed to be around here. Yes, so you see codominance in complete dominancy. No, that one is fine. In complete dominancy here we say that they say that this word, you see this word, it's what they said, intermediate. You see, intermediate, and then the title here is incomplete dominancy. And then when you talk about codominancy, it produces a mixture. It produces a mixture. Both allele are, yes, expressed. So it means that here you produce a mixture of two colors. So that's why I'm saying that incomplete dominancy is the best correct answer. Then we go to the next question. We are saying that the fluid fluid of the nucleus where free nucleotides are found, we call it a nucleoplasm. Plasm means fluid, and then nucleo from the word nucleus, therefore is the fluid of the nucleus. Then you are saying that the tangled mass of chromosome located within the nucleus. So we call this one chromatin network. So here is chromatin network. If you have any challenge, we can just show you. I can just show you on the distinction material. As I told you that distinction material, there is no question which cannot be answered by that book. Yes. So I wrote that book with the great time so that it can tackle any question they will set before and after you have written. So if you use distinction material, now I'm talking about those people who will be watching, not you now, definitely you will get a distinction. All right. Division of the cytoplasm, we call it cytokinesis. We talked about it yesterday. Say that and they're saying that the name of the, someone is saying, they're saying that is talking about the chromatin network. We talked about the chromatin network. It is here. We talked about it here. You see, entangled. You see, this is what you call chromatin. So it's a chromatin network. Yes. So that's what they are talking about. Here is the thing. Chromatin with chromosome consists of DNA and protein. So that is what they are looking at. This entangled. Yeah. That's what they are talking about. Ne? Yes. All right. We go to the next question. The name of the X and Y chromosome in male, we talked about this, gonozome. So I think we are clear there. Someone is giving me the answer already. Gonozome is correct. Let's go to, I hope you guys, you got 10 out of 10. If you got 10 out of 10, congratulations. If you lost one, at least you tried. Yes, you're still in the range of 95%. Genetic disorder characterized by chromosome mutation. I don't know how many people got this right. Let me just say it because I have no choice, because I have to tell you what is the most correct answer here. They're saying that genetic disorder caused by chromosomal mutation. The answer is none. I'm sorry if you wrote something or both. Ne? It's none. Yes. Why none? Because these are gene mutations. They are gene mutation, not chromosomal mutations. When you talk about the chromosomal mutation, we are talking about the whole chromosome changing. Therefore, we go back to Down syndrome. And if you check on Down syndrome, sometimes we call it chromosome mutation, sometimes we call it chromosomal aberration. So when you look at Down syndrome, that's where you can find chromosomal mutation. Let me show you what I talked about Down syndrome. When you come here, yeah, I think I didn't indicate it here, but it is chromosomal mutation. Yes. I don't remember where I wrote it, but it is there, whereby you talk about the whole chromosome is changing. Yes, when you change the number or the structure of chromosome, then we call it chromosomal mutation. Sometimes we call it chromosomal aberration. Yes, aberration or chromosomal mutation. But if you just change the base, then we call it gene mutation. So these two are gene mutation because the as a result of exchange of a base, a specific base. Then we are saying that importance of meiosis is both A and B because there is formation of gametes and then having the chromosome number. Yes, so this is both A and B. And then I'm saying that organelle where DNA is found, both A and B. We talked about this, and yesterday I was tricking you, and then someone got it right. I asked a question that give me any other place where you can find DNA other than a chloroplast. No, other than a mitochondrion. And then someone saying that chloroplast. And then the person couldn't tell me that nucleus. Ne? Yes, so but what I wanted to tell them that think about mitochondria and think about the chloroplast. You understand? Good enough that this question came, and I hope that person tried to get the answer right. So the answer here is both A and B. Don't forget that in paper 1 I told you that you must say that both A and B, not A and B, not both. If you wrote both, I'm sorry. If you wrote A and B, I'm sorry, you did not follow the instructions. However, maybe with the mercy of God, then maybe they might correct it, but you have to follow the instructions. Now I'm talking about those who are going to be writing the next paper. Then we are going to question 1.4. I hope guys you got 6 out of 6. Ne? Yes, question 1.4. Oh, blood group. We talked about blood group yesterday, but let's see what was here. Name the type of diagram represented above. This is pedigree. Pedigree. Yes, so this one is correct. We talked about it, and someone was asking what's the difference between pedigree and the phylogenetic tree. So now we are clear there. Give the number of the allele that control blood group. We talked about this yesterday. We said there are 3 alleles. Alleles. Yes. Alleles or alleles, whatever case you call it, is fine, man. Just a moment. Sorry, this another Samsung is here disturbing me. Just a moment, guys. I don't want to this Samsung. Yes, I'm back. I'm back. I didn't want this Samsung. Okay, so we are saying that the diagram below represents the blood group in the family, or blood group O. Okay, sharp. Then we have name the type of diagram represented above. We said that the answer here is pedigree, so it is fine there. Then they are saying that give the number of allele. We said it is 3 alleles. How many generations are represented in the diagram? We said we count 1, 2, 3. So the answer becomes 3 generations. Yes. They are saying that Lena's genotype is this. Lena's genotype is A. Yes, you remember what we talked about? Okay, now Let's go to the question. State the possible phenotype of VUSI. Possible phenotypes. Phenotypes or genotypes. They want the genotype of VUSI. So if you are talking about the genotype of VUSI, where is VUSI? VUSI is here. But now, if Lena is A, small a, VUSI, this person I don't know is small I, small I. Remember, I could ask a question that how is a person of blood group A and a person of blood group B can produce a kid of blood group O? Then I said yes, because if they both have heterozygous, they're in heterozygous state, they have the recessive allele, therefore they can produce a kid of blood group O. Therefore, how many marks they give it to VUSI is 2 marks. And they're saying possible. Therefore, VUSI can be A, small I. Yes, that's number 1. VUSI can be B, small I. VUSI can be I, small I, like that. Whereby if this combine with this, you produce O. If this combine with this, you produce O. If one of these combine with this, you produce O. You see that any of that can lead to VUSI. So they want 2 of them. If you wrote this and this, cool. If you wrote this and this, cool. Any of this, they wanted 2 of them. So I think, yes, you will get it right. Ne? Yes. Someone is saying that 4 generations. I don't know where did you get the 4 generations, but here is showing you 3 generations. Ne? Yeah. All right. Then they're saying that give the genotype of Bob. Where's Bob? Bob. So if this person is O, if this person is also O, definitely there's no way how Bob will be A or B. It will be O. So they're saying genotype of O, of Bob. Therefore, Bob is gonna be II. So I think that one is also correct. Give the name of the individual who displayed the codominancy. Codominancy means that you have blood group A and blood group AB. That's what they are looking for. So the first person I can think of here is you see here now, this person is blood group O. You see, blood group O. But the kids they produced is blood group B and blood group A. Definitely Anna is blood group A, B. Do you understand? Yes. So this one is here, can combine with this to produce B, to produce A. Sorry, this can combine with this to produce A, and this can combine with this to produce B. Therefore, Anna is definitely answer. But here it's showing you, so it's Anna. Is it Anna? Anne. Ne? Yes. So here they're showing you 2 marks. Then you have to look for another one. If not, if they don't want one, you have to look for another one. Another one, do we have another one? We can get another one just in case that Lena and Anne are kids of these 2. You understand? But Lena is having blood group A. And then Anne is having blood group AB. Definitely, if this is blood group A, ne, and this is blood group A, how will they produce this? Maybe one is blood group AB. You understand? Whereby this combine with this to produce blood group AB, which is Anne. You understand? What if? Because they said, did they say give the name of individual which displays codominancy? So the Anna is the perfect answer. However, based on this and this, so either this, because I can have this here, or I can be, maybe this can be A, because if I combine this with this, still I'll produce this person. You see, if I combine this and this, I'll produce this person. So is either any of this is either A, like that, and then B, like that, or it's like this. Either of these could be. So it means that based on the analysis, deep analysis, not just easy analysis, it could be Curis and this, one of the 2, not both. You understand? Not both. Both can't be AB. One of them must be AB. Then the other one has to be a heterozygous. You understand? Yes. So one of them could be. So the next answer, if an is the obvious answer, then the person who wrote either this or this could be also another correct answer. Someone is saying Vusi. What about Vusi? Let me see. Vusi is having blood group AB. No, there is no way how you can get blood group AB when someone is having blood group O. You understand? Because you don't have the second recessive allele. You understand? Yes. Yeah. Someone is telling me name. Thank you very much. Yes, I know that you are saying name. This is the obvious answer. Yes, it's a name. But don't forget that this could also be one of them correct. So the memo could have an extended opportunities after the discussion. However much, they could not see this maybe before they do the memorandum. But when you go to deep analysis, then you find out that this and this, there could be some student who wrote it and it could not be discredited. So should be credited if it results in that. You understand? Yes, obvious answer is that. However, even those, either of this could be an answer. We go to the next question. You're saying that the next answer is question below is a representation of protein. Okay, protein synthesis. We know that what is here is being protein synthesis. So now remember guys, we discussed about this and I told you that please don't make a mistake to go without knowing this. I told you that one of the paper, depending on which section, which paper they have brought, for example, if set 1, set 2, set 3, whatever, if it happens, one set must have at least transcription, maybe another must have translation, another must have DNA replication. One of it must come. And then that's what I said, and say that please don't go to the paper without knowing it. All right, let's continue. They're saying that 1, 2, 1, okay, sharp. Let's go to the question and then we see Z. Z. What is Z? Name, identify Z. Where is Z? Am I lost? Where is Z now? Oh, sorry. I couldn't see Z. The process Z. Process Z is, what is this? Remember, this is uracil. And then this is DNA. Definitely, this is transcription. Transcription. Remember, transcription is the formation of messenger RNA. So, molecule R. Where is R? Oh, molecule R is messenger RNA. Then they are saying that organelle Q. Organelle Q. Where is this? Where is organelle Q? Okay, organelle Q is the ribosome. Yes, ribosome. So guys, I used to tell you that whenever they think of organelle, think about two things, either nucleus or ribosome. So that the two organelles we talk about here, don't have any other organelle. Give the collective name for nitrogenous base O. This is O, ne? O is gonna be anti-codon. Remember we said that code for DNA, codons for messenger RNA, anti-codon for transfer RNA. And I told you how to identify that this is a transfer RNA. Just write a T, transfer RNA. ZT, and then you call it messenger. When you see ZT, ZT, so it's a transfer RNA. So the codes which found there, we call them anticodons. Determine the sequence of the nitrogenous base at S. Where is S? Oh, it's here. and remember this is, let me check, is this is T, and also let me check is T. This one, it has T, even this one has T. Therefore this strand is for DNA. Therefore I've just said T goes with A, and then C goes with G, and then A goes with T. So basically that's what you're supposed to write there, here, the sequence. Which strand 1 or 2 is used as a template for formation of R? So we come back to R. So we have to mirror this. So this A goes with the T, U goes with the A, C goes with the G. So I have to look for what is this. We can continue. TAG. So strand 1 is the one which is used as a template so that this can be formed. Some people could have, maybe they have written strand 2 because this thing is facing that side, but that's just a confusion. It's just a confusion to see that some people are off. All right, so the answer here is gonna be strand 1. Yes. Someone wrote spinal cord. Wait. Okay, sharp. Someone said it's a spinal cord. Aibo. No, man. You guys, you're acting in the paper. All right. AGT. Okay, sharp. Then they're saying that which amino acid 3 or 4 will be brought to area P. Area P. Where is area P? Okay. Guys, there is something which was hidden here. Let me show you that hidden part of it. And I hope you guys got it right using the elimination method. Okay, all right. If we follow this, following this, yes, I hope we can get it. But if you just follow what is here, because there are 3, and here you have the bases, so definitely this goes with A goes with U, because this is transferring A, U goes with the A, then this goes with the G. So UAG, so this one is gone, so we don't talk about it. G goes with the C, G, C, and then A. CCA, then this is gone also. Definitely this one will come there. You see? So you can see how you can identify it correctly. So basically here we come here and we say that amino acid number 4, amino acid number 4. So that's the correct answer there. Then they are saying that name the type of sugar that forms part of structure molecule R. Part of sugar molecule R. Part of sugar. Name the type of sugar, not part. Molecule R is messenger RNA. So here they are talking about sugar. So it's gonna be ribose. Don't say ribo. No, it's a ribose because they are looking at the sugar. So guys, I think you got 50 out of 50. Congratulations if you got it. Yeah, so it looked to be a little bit simple. Is there anyone who got 50 out of 50? Let me know in the comment section. Someone is saying 1.5.2. The collective name of nitrogen. They say the collective name of nitrogenous base. The collective name of. They are not talking about what is this. No, they are saying nitrogen. The key point here is nitrogenous base. So someone is talking about tRNA. tRNA is only this. but they're talking about the collective name of these 3 bases which are found on the tRNA. Therefore, the answer becomes the nitrogenous bases. I think I'm clear there. Let's go to question 2.1. Uh, 48, 35, 40, 18. Still 18, you can still do it. You can still do it. Let's go to section B and then you see how much you're gonna collect with the 50. Yes. Section B. Then they are saying that name the structure. Name the structure. Name the structure. So here is, I told you about it. Yes. Name the structure. B. What is B? We come here, we look for B. Is a century. Remember yesterday I told you that please turn it so that it runs east to west. Is it east? Is it east way? Is it west? Is A? I'm not a geographer, guys. Don't quote me wrong. I think it's east this side and then west this side. I hope so. Yeah, so it must be running east west or in the horizontal way so that you see it clearly. For example, it's supposed to be like this so that now you see it must be running in that direction so that you see the actual phase. Yes, so I hope you guys you did it and identify the phase correctly. Okay, let's go through it and then we find out. Okay, now we are saying that B is centrioles, spindle fibers. These are homologous chromosomes, but I don't know whether they're asking you. Okay, spindle fibers, we are done. Identify the phase in diagram 1. Diagram 1, what is happening? You are seeing here crossing over is taking place. There are 4. It is prophase 1. Prophase 1. Give 3 reasons for your answer. Why are you saying it is prophase 1? First of all, you are forming here a bivalent. Chromosomes are under crossing over. Homologous chromosomes are under crossing over, you see. Yes, that's number 1. The nuclear membrane is starting to disappear, you see. Nuclear membranes are starting to disappear. The centrioles are moving to the opposite poles. So you can talk about that. Yes. Yeah. If there is any other, it could be accepted. I don't know what you wrote, but as long as it is correct. Describe the process that takes place at A. They're talking about the process of crossing over. Homologous chromosomes come together or align together, and then chromosomes of homologous chromatids, chromatids of homologous chromosome, they cross over at a point called chiasma. Yes, they exchange the genetic material, and then they split to form 2 genetically different chromosomes. So basically you just explain the process of crossing over. As I say that you cannot go to the paper unless you are insulting me when you don't know the process of crossing over. Yes, I talked about that. I said don't go to the paper without knowing the process of crossing over. Don't go to the paper without knowing the metaphase. Don't go to the paper without knowing anaphase. The rest you can skip, but those 3 don't make a mistake to go there. I think those who read it is not. Someone is telling me that spindle fibers are formed. Spindle fibers are formed, they are moving, centrioles are moving to opposite poles. Spindle fibers are not formed. Spindle fibers are forming. They are not yet, you see. Yeah, it's also correct. It's also correct. Spindle fibers are forming. Yes. Yeah, because if they are formed, they have already formed like this. So they are forming. Remember, in prophase, they are in the process. Everything is in the process of forming. All right. That's English, but in most cases, we also mark English. All right, let's go to the describe the process. We are done with that. Identify the phase represent diagram 1, diagram 2. This I said you swap it because they're in pairs, therefore it is meiosis 1. Therefore, because they're in the middle, you see they're in the middle, you swap it, you turn it, yes, as I told you, east to west, therefore it is metaphase 1. So the answer there becomes metaphase 1. They're saying that describe the difference in the event that takes place in phase mentioned above. Describe the differences in the events that take place in the phase mentioned in A and the same phase during mitosis. Or they want the differences between this phase, meta phase 1 and meta phase. Remember, mitosis doesn't have meta phase 1, meta phase 2. Here, chromosomes align along the equator, yes, in homologous pair randomly. You see, randomly. Chromosomes align along the equator in homologous pair randomly. While here, individual chromosomes align along the equator singly. In actual sense, they're asking you meiosis 1 and meiosis 2 of the same phase. Remember, we said that meiosis 2 looks like mitosis. Yes. So basically, chromosomes align along the equator randomly in homologous pair. That's the key point. Homologous pair. Yes. While here they align along the equator singly. Yes. So they're saying differences. They're not talking about tabulate. So they don't need you to write the table or to draw the table. Describe the results at the end of meiosis if chromosome D fails to separate. Chromosome D. Okay, let me just turn it so that you see it clearly. What happens if chromosome D does not separate? So basically, this is not a human cell. So don't talk about 47 chromosomes. Here, what you say, okay, definitely it's going to be like this. Technology is jamming. Okay. Still jamming. Okay, it's supposed to go that way. Just a moment, just 2 seconds. As it's back. It's back. Okay, now you're saying that it's gonna go through like this, and the whole thing, this becomes one cell, and then this one becomes another cell. So it means that what you describe here, say that one cell we have 1, 2, 3, 4, 5 chromosomes. Yes, one cell we have 5 chromosomes, while the other ones instead of 4, because it's supposed to be 1, 2, 3, 4. Then the other one we have 1, 2, 3. So we have 3 chromosomes instead of 4. You see? Yeah. So basically that's what they are looking at, or they are looking for. Yes, that's what they are looking for. So that's why they ask, describe the result at the end of meiosis. At the end of meiosis. Yes, if D fails to separate. Yeah, so it means that one cell will have an extra chromosome, 5 chromosomes, while the other ones will have 3 chromosomes instead of 4, and then the other one will have 5 chromosomes instead of 4, meaning that you have one extra chromosome, and then the other one will have one short of chromosome. That's it. Then number 2, those people who wrote about 46 chromosomes, they didn't talk about here that this is a human what? They didn't talk about that this is a human cell. Yes. Number 2, look critically here. Let me show you something here. If if this cell, these two cells, this one is gonna receive x, 1 2 3 4 5, ne? And then this one is gonna receive 1 2 3. So the fourth one is gonna be empty. I see. Yes. So it means that now when this one divide, this cell is gonna have 1, it's gonna have 5 of them, and then the other one, when it divides, they're gonna be 2 of them, but each will only have 3. While this one, each of them, remember it's meiosis 2 now, each of them is gonna have 5 because it's supposed to split equally. Each of it is gonna have 5, and then this one, each of it, because there are 3 now, this one is 0, it's gonna be 3. You see, it's gonna be 3. That's another option, but still it gives you 3 here, 5 here, 5 here. 2 cells will have 5 each chromosome, while instead of 4 each, and then this one will have 3 each instead of 4. I think I'm clear. If you wrote it in that way, still they will credit you. Describe, so we're done with that. Describe the process of DNA replication. Perfect. I think I was clear here, emphasized, and even say that if you could see my face. All right, DNA replication is a process by which DNA makes identical copy of itself. You understand? Yes. So you now tell us that DNA double helix unwind, weak hydrogen bonds break, then weak hydrogen bonds breaking is the same thing saying unzip to form 2 templates, each. So to form 2 separate strands. What we want to hear the word separate. 2 separate strands. Each is acting as a template for formation of new strand using free-floating DNA nucleotide. Remember, if it is transcription, messenger RNA nucleotide. But here we say DNA nucleotide from the nucleoplasm, whereby a complementary strand is formed whereby A goes with the T and the C goes with G. Is it clear? And then 2 new DNA strands are formed. 2 new DNA are formed, each with 1 original strand and each with 1 new strand. This process is controlled by enzyme. It could also be right. So this question has more than 6 marks. They have given it to it. So it means that the only credit 6, and then we write maximum, meaning that you have reached the maximum, therefore you got it. People are always asking me about the spelling. Okay. Tando is asking me about the spelling. Tando, what if I call you Tondo? Will you feel good? Instead of calling you Tando, I call you Tondo. I replace A with O. Will you feel good? Will you be the same person? If you are not the same person, why do you want to make mistakes with the spellings here? All right, it will depend on them, whether it means something else or it's the same thing. But we always encourage you to write correct spellings. All right, let's continue to the next question. Read the passage. Gene, okay, code for blood clotting in humans. This gene made up of that 163 amino acids. The mutation occurred and result in amino acid 123, 128 sequence. This changes to this, and then this changes to this. Mutation has been transmitted as autosomal. Yes, it has been transmitted as what? Autosomal. And they are saying that they're saying that the mutation has resulted in a resistance of warfarin drug. Warfarin drug in human. Warfarin is used to treat prothrombosis. I'm used to the process of clotting. Prothrombin, fibrinogen, fibrin. Okay, prothrombin is the one which produce thrombin. So here thrombosis is the disease. Okay, treatment of thrombosis. Thrombosis results in the formation of blood clotting in the arteries, and then this causes the thin of blood to break down and the blood clot. Name one piece of evidence in the information that show that mutation for this gene occurred in DNA molecule. How do I know that mutation occurred in DNA molecule? Check here. Yes, it changed from here to this and here to this. Where do you find the T? Definitely find T in DNA. Therefore, that is the correct answer. So you talk about that, yes, the sequence change of this, change to this, and also this change to this. This shows that it was DNA. You understand? Yes, because DNA is the only one which has the thymine. Yes. Then number 2, they're saying that how many nitrogenous bases code for this gene? Hey guys, I talked about this. I even showed you how to calculate these nitrogenous bases. Remember, each nitrogenous base, each amino acid is coded by 3 nitrogenous bases. Therefore, I will say that. Yeah, someone is saying it has thymine. Yes, correct, because of this thymine. Yes, presence of thymine. Yes. Okay, saying that how many nitrogen? You're gonna say 163 amino acids times 3. So the answer you get is what you call let me see, how much is it? Okay, you calculate. Let me know the answer in the comment. Yes, describe what is meant by autosomal dominant allele. Autosomal, it means that is a non-sex chromosome. Non-sex, it is not carried on X chromosome. You understand? It is not carried on X chromosome. This allele is not carried on X chromosome and is dominant. Yes, it is dominant. That's what it means. It's an autosomal dominant allele. It's dominant and is not carried on X chromosome. So this disorder is caused, or this condition is caused by a dominant allele which is not carried on X chromosome. Yes. Then they are saying that the table below shows the amino acid and their corresponding codons. Hey guys, when I was trying to explain to you these mutations, I told you that you need to take note of this. Yes, I said it. The table below shows the amino acids and their corresponding codons. So meaning that this is messenger RNA, because they said that codons. They didn't say transfer RNA or anticodon. How will the mutation on this result in a resistance of this? Check 5 marks. What are you supposed to do now, man? What you are supposed to do here is you come back here and read here. A mutation occurred affected the amino acid this, and the sequence of this to this is changed, becoming this. This mutation has been transmitted as an autosomal dominant characteristics throughout the generation. This is the problem now. It has formed another protein. You understand? But how do I know that it's forming another protein? Okay, now the first step to score those 5 marks, you had to tell us what we started with and what we ended with. So we started with G, T, G, which changed to C, A, G. You understand? And then you have T, A, T. Someone said that is 489. Here is 489. Guys, if it is correct, you calculate it, then that's the perfect answer. Then you have to tell us that this one changed to from what? Okay, let me just make a big space so that we can know exactly what we are trying to show you. Okay, this is the first amino acid, this one, So first one, and then the second one, let me write it here. It is T A T and then T C T. But now, what do these ones indicate? They're saying these are codons. Yes, so I don't know, I can't read it here unless I change this. So change it. So this is the first mark by changing G A C, and then this one is G U C. What does GAC mean? GAC, let me first remove this so that I see clearly. GAC is leucine. It indicates leucine, this one. And then GUC indicates glycine. Yes. Now what about this? Let's convert this. This is gonna be it's got A U A, and then this one is gonna be A G A. So what does this one indicate? This one indicates this one indicates AUA. AUA is phenylalanine. Phenylalanine is this one, and then this one, what does it indicate? AGA is arginine. Guys, these are just names, ne? Yeah, you don't need to know them by head, ne? Me, I'm saying them that arginine, what? Yeah, because we find this in biochem, ne? Yes, in biochem. So this is it. These are the amino acids, which you know that, yeah, the essential and non-essential amino acids. So as long as you write what they gave you. All right, now we have translated. You see, we have what? Translated. Here we will get a mark. We will get a mark. It's 1 mark, 2 marks. Then also changing this is another mark, 3 marks. Then changing this to this is another mark, which is 4 marks. You understand? But now put them in words. The codon, the first codon, AGA, ACGS, CGA was changed to ACG, cool, which indicates that this was changed to this, okay? And then this produced leucine. It produced glycine instead of leucine. So you have changed the amino acid. And then also this one, the second phase, we changed the amino acid from phenylalanine into arginine. So different amino acids will be changed. This will be changed into this, and also phenylalanine will be changed into arginine. You understand? Which makes a different protein. It won't be the same protein because these codes which change, they change the amino acid. So therefore a different protein is formed. That's what it means. So you guys were supposed to show us that this changed to this changed to this, meaning that new amino acid is formed. So it will be coded for glycine instead of leucine, and then here it will be coded for arginine instead of phenylalanine. Therefore a different protein will be formed, and then this protein is resistant because they said the mutation has resulted in resistance. Yes, this protein which has been formed result in the resistance to this drug. So basically that's what they wanted. I explained it yesterday, but now when I'm seeing this question, the way how they try to trick it, when I was explaining it, I explained using one example, and I told you that don't make a mistake to go to the paper without knowing this, because I know they're gonna bring it, and I know that people are going to have a challenge to explain this. And these people, you know what they do, that if they find out that this question is hard for these students, they keep on bringing it. I don't know why, but they keep bringing it. Maybe they want to make sure that everyone knows that concept. But if they see that now you can know this one is easy, they don't bring that one anymore. They bring more which is a little bit difficult to see that, yeah, you can apply. But I hope you guys, you got this question correct. Let's go to the next question. Let's go to the next question. They're saying that they're saying that the effect of this mutation has explained the effect of this mutation has with the prothrombic. This one you just come back to the extract and then you read here. The mutation has resulted in a resistance, you see? Yes. And then in the human of treatment of used to treat this, the thrombosis result in the formation of blood clot in artery. So this will result in the, yes, you see, the effect of this mutation on human with this. So meaning that it will result resistance. First of all, the mutation will bring about resistance. Number two, it will also bring about this formation of blood clot in the artery, you see? Yes. And then also will cause this to break down, you see? So basically, no, this one is out because this is a drug, né? So it means that it will not allow this blood to break down, the clot to break down, because now it is now resistant. So meaning that the blood clot will remain there. Why? Because of the mutation which has developed. Therefore, you talk about the resistance, and then you talk about what this you see here, this formation of the blood clot in the artery. Therefore, these clots will not be broken down because of the resistance which has been developed. So basically, the answer was supposed to be obtained from there. Hemoglobin. Someone is saying that if I could say that it's okay, has deleted it. Okay, it's fine. So guys, I think this question was one of the challenging questions, just challenging questions, and I knew that it's coming. And then when I was emphasizing it, yes, I tried to see that everyone can pass it. Yeah, we go to the next question. That is question 2.4. Question 2.4, we are saying that polydactyl, these are the people who have extra finger. Maybe they have 6 instead of 5, ne? They have 6 instead of 5, lead to extra finger on extra finger or toys. It is caused by a dominant allele. This word, you remember I told you guys when you talk about dominancy? Yes, a man who is heterozygous for this, meaning that is capital R, small r. Yes, because they said use R, ne? And a wife who is not, meaning that it's a recessive, so is homozygous recessive. Use the letters R and the RR, do the genetic cross to show the percentage chance that their children will have polydactyl. The percentage chance of having the polydactyl. So here, what are you going to write? So it's simple, guys. Here is very simple. Let me just talk about it here. P1, F1. I told you about that, and I said we gave you a mark. Then we say phenotype. I think you guys, you wrote it that. You go back to the question, you find the phenotype. They say that homozygous, a man who is having the condition, a man with polydactyl. Yes, polydactyl. So you're gonna have to say a man with, yes, polydactyl, crossed with a lady without polydactyl. Yes, meaning that here I'm gonna have a lady, wife with no, yes, here with, yeah. So you finish. Then here is genotype, but they say the man is what? They say the man is heterozygous. Therefore, I'm gonna say capital R, small r, crossed with a lady who is recessive. So here I'm gonna say meiosis occurs, and then I'm gonna say capital R, small r, small r. I say these are gametes. If you didn't circle them, it's fine. If you circle them, it's fine. Yes. Then now you can use the Punnett square method, yes, or you can cross. Either way, as long as it's correct, but you write the word fertilization if you use the Punnett square, or you write fertilization here, fertilization here, and then you say C, and then you start capital R small r, this with this, capital R small r, this with this, small r, then this with this, small r. So these are offsprings, you understand? Yes. So basically this is what you're supposed to do to get it right. But now we come here, we mark. If you don't know, if you got this wrong, then automatically we shall come here, we say fertilization, it will give you a mark. And also writing this, you see now you have 3 out of what? Out of 6. But if you got all these correct now, but the question is asking you, there might be a compulsory mark. Let's find the compulsory mark here. They're saying that use the letters, do a genetic cross to show the percentage chance. Yes, the percentage chance of so percentage is a key point. Is a key point of having the kids with the disease. So basically we have 2 people normal is caused by a dominant allele, 2 people with the disease, and 2 people without. So basically it's gonna be 2 out of 4 times 100, which gives you 50%. You see, this is a key point. We mark you. So we're gonna mark you here, we mark you here, we mark you here, but we're gonna first count this. How many marks are there? How many marks we are given? Yeah, we're gonna give you this one plus any other 5. Yes, or it will depend on, but this is the compulsory mark which they're gonna give. Someone is asking me about agriculture science. So many people asking me about agriculture science. Guys, I told you that unfortunately this year I'm not sure that you're gonna work with the agriculture science, but next year 2024 we will work with the agriculture science. Yes, it's because currently we don't have a school which has which does the agriculture science in this area. Unless you send it to me, you scan it, you send it to me, then we'll do a memorandum for you. Yes. All right, yeah, saying that below shows the graph below shows, I think guys you got all the marks here. I'm skipping, I'm sorry. Okay, so we are done with that, né? So let's go in summer squash plants. White fruit is dominant, né? So we need to know that white is dominant over yellow, and then round is dominant over oval. So this is dominant, white over yellow. Okay, now let's start. Summer squash plants that are homozygous for white and round is crossed with plant that is homozygous yellow. Okay, sharp. Homozygous, remember yellow is recessive, né? And then this one is white is dominant, and they are saying that dominant around. Oh, dominant over crossed with recessive. Okay, give the genotype of parent 1, parent in the first generation. Okay, parents in this case they say that white, therefore capital B because they said homozygous, and then white, and then round is capital D because they said homozygous. So that is the first, and then the first mark, and then the second mark could be is the crossed with the homozygous yellow. Yellow is, we say, the small b, and then oval is small d. So that's the second mark. So that's what they are looking at here. The phenotype of F1 generation, because all of them are dominant, therefore is going to be capital B, small b, capital D, small d. So the phenotype both is going to be white and yellow. So it's going to be white, correct, and over. Did I say? Not over, man. It's not over. The one which is dominant, because I have capital D. Capital D here indicates round, right? Round. So it's going to be white, a mark, and round, another mark. Then they are saying that two plants that are heterozygous for both characteristics were crossed. Okay. What if I use, someone is asking me, what if I use X and Y? Guys, I told you that in this generation, in this, you only use X and Y if they have told you to use X and Y. So they didn't tell you that this is X linked, né? So the answer will be wrong. But if you wrote this, and this, then automatically you will get the answer. Those remarks, and if you got the percentages 50, you'll also get another answer. At least maybe you'll get 4 out of 6, né? At least you did not come out with a 0, né? Yes. They're saying that the two plants that are heterozygous for both characteristics were crossed, meaning that I have capital B, small b. I have capital B, small b, capital D, small d. So this is what I have. So if they are crossed, all possible genotypes of the gametes. So here I'm going to find out the gametes. This gonna go with this, as I told you yesterday. This is exactly the same thing I told you yesterday. This we're gonna go with this. It's gonna be capital B, small d. Then this with this, it's gonna be capital small b, capital D. This with this is gonna be capital B, small b, small d. So these are the gametes which are possible. So 4 of them, maybe 2 mark by each. So you get that correct. Then we go to the next question, guys. If there's any question I've skipped, you let me know, né? Sometimes I skip when I don't see. All right, how many plants in, okay, so how many plants in the next generation are likely to have yellow and oval fruit? How many plants in the next generation are likely to have yellow and oval fruit? Yellow and oval. So we come back here and look at for yellow. Where is yellow? Yellow, 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 and oval. Yellow. Okay. Now they're saying that, okay, both were crossed, heterozygous. So automatically, when you cross, you'll find out that because both are homozygous recessive, meaning that they are going to be small b, small b, small d, small d. So the answer here is going to be 1. One plant. Yes. Or maybe 1 out of 16. Yes. So 1 plant, or 1 plant out of 16. I think let me just show you here from the distinction material what they are trying to show you without doing the genetic cross. Yes. This is what I'm saying. You see, they are looking at this. It's only one which has both letters when they are homozygous recessive. So basically that's what they are looking at. When you come back here, that's what they are looking at. It is small b, small b, small d, small d. That's what they are looking at here. Capital small b, small b, small l, small l. Yes. So and the percentage here, if you count all of them, they are 16. Therefore it is 1. It's only they are looking for this. Yeah, one individual, or 1 out of 16 individuals. So I think I'm clear there. Then we go to the next one. Yeah, someone got it right. I think, guys, you are collecting marks now, né? You are collecting. Give a possible genotype of both parents that must be crossed. A farmer wants squash, okay, that way, white and oval. Yes. So the two, if you want white, basically if you want all of them are supposed to are white, which is capital letter, and then oval is a small letter, meaning that the resultant must be capital B. For example, it must have a capital B, and then it must have two small d's, because they say the oval, and the oval is recessive, you see. So basically the genotypes of the parents must be this: capital B, small d. That's the first person. And then the second person can be capital B, small d. That's the second person. Or the first one can be like this, and then the second one can be capital B, small b, capital D, because this B will be somewhere. Yes, so basically if you wrote this, they will give you a mark. I think they want to, let me see, possible genotypes. They want to this with this. Yes, or this with this is cool, or this with this is also cool. So yeah, that's what you were supposed to write, so that at least you get the correct mark there. So the options there, if I have not mentioned another option, then automatically it means that it will be marked correct. Who got 50 out of 50 in section question 2? 50 out of 50. Let's go to question 3. Someone is asking me, you describe, I don't know, what was that question? I don't remember what question was it. Okay, let's go to the next question, and then they're saying that the graph below shows the results of artificial selection for protein content in the meal plant over 50 generations. Describe how the farmer did artificial selection. Here you just had to explain the artificial selection. Remember, in artificial selection, you identify the character, describe how this farmer did. So he identified, you see, they identified the character of producing protein in the meal, and then after that he identified those plants with that character. Yes, then he crossed those plants with those ones which have that character, which have that character. He crossed them. Yes, then after that he selected only those which produced more in the first generation, and they were allowed to cross-pollinate, because they don't do sex here. It's close pollination. It's pollination. So he cross-pollinated those which could produce large number of protein. You understand? And then at the end of the day, he found out that the plants were able to produce large amount of protein. What was the average percentage of protein content in the meal in at the 15th generation? So here you just had to draw a line touching there, and then you draw a line touching there, and then you find out maybe 12.8, and then you put these units. Kernels. Yes, so that becomes the answer. And then someone is going to ask, what about 12.89, or what about 12.9? Yeah, they will give a range. Yes, someone got 38. That's still good mark. 42, still good mark. Is good mark. All right, guys, thank you very much for getting those good marks. At least you are promising. But by how many times did the average percentage of protein content in meal grain increased between this, meaning that they are looking at for percentage increase. They have brought the question again. By how many times? By how many times? By how many times? Okay, the average percentage of protein, yes, increased between 40 and 45. I come here, I find what is final. The final is 20. 20 minus the initial, which is 14. So you get how much here? You get 6. 6 divided by the initial, which is 14, times 100. So the answer you get here is the number of times it increased. Yes. In terms of percentage, saying that by how many times did the average percentage of protein, yes, okay, you had from 14 to 20. So someone might have a different version. So you guys, you have to calculate for me, and say it's only 1 mark, so you how many times from 14 to 20? Basically, that's what they are looking at. So when you do the percentage increase, then you'll be able to know how many times. And then here, you guys, you calculate and you get the answer. Yes. Describe one way in which the process of artificial selection is different from genetic engineering. We talked about this. Artificial selection, there is no manipulation of genes. You only have to select those organisms which are desirable by the human, and they are given a chance to reproduce. While in genetic engineering, you don't have to cross these organisms. You just have to select using, there is manipulation of genes. That's why it's called genetic engineering, manipulation of genes. While here, it's only you cross the organisms. You don't touch the genetic composition of an organism. Yes. Number 2, this one is a little bit of expensive. This one is a little bit of cheap, in which this process they differ. Okay. This one takes a short period of time, while this one takes generations and generations. Yes. Someone is saying 12.5. Someone is asking me, is it fine if I don't put the units there? Hey guys, those are the discussions we do, or the discussions they do when it comes to the discussion. Should we correct this? Should we not? So basically, what you need to do is you must not always forget about the units, because the units are very, very, very important. All right, let's go to the next question. Darwin's theory of evolution by natural selection. I explained this, and I told you that unless they bring it as like this, then I also explained to you what you're supposed to say. Natural selection, you talk about there is variation among organisms, those which are favorable, those with favorable characteristics, because they didn't give you here an example, so you don't pick an example. Those with favorable characteristics and those without favorable characteristics. Those with favorable characteristics, they reproduce in large numbers. They survive and reproduce in large numbers, and they pass their genes to the next generation. And those without favorable characteristics, they die in large numbers. This is called natural selection. So those which pass their genes to the next generation, yes, they reproduce in large numbers, and then after here you also supposed to talk about, just in case, because it's 7 marks, maybe you can talk about natural selection is what is a survival of the what of the fittest, which organism is best suited. Sometimes when students, they are trying to define this, because this is a block question whereby it is no example, no what, you just have to explain just what you know. Yes, you on natural selection. So basically, this is what I'm trying to say. Yes, I'm almost there. Yes, here. Natural selection. Yes, it's natural. Organism produce large number of offspring. There is a great deal of variation. Some have favorable characteristics and some do not have those. When there is a change in environmental conditions, yes, if there is competition, the organisms with favorable characteristics will survive, and then those with unfavorable characteristics will die. Organisms with those which will survive will reproduce in large numbers, thus pass their alleles to the next generation, to their offspring. So basically that's what they are looking at. Just describe the process of natural selection. That's it. Then we go to the next question, guys. That's what they wanted from there. 3.33, investigation was done to determine the relationship between height and the head and the bite force. The height sorry guys, there was some technical problem with the sound, but now it's back. Okay, they're saying that what I was saying that the investigation was carried out to see the relationship between height of the head and the bite force in lizards. Ne? Yes, the bite force in lizards. Scientists collected all of that, so you are supposed to read there. Ne? So now, what is the first question there? What is the independent variable? So the species will determine the head and also determine the force of the bite or bite force. So the species becomes I, D, X, Y. I told you that. Ne? So this becomes the species, becomes the independent variable. And the independent variable, then the bite force. The bite force. Yeah, someone can ask me that, what about that? Height determines the force of the bite. You understand? How much force do you need in neurons? Yes, the height will, because as the height increases, the force also increases. Yes, but what determines this is the type of the species. So yes, if it was this and this would have been the independent variable, dependent variable. But now we have this which determines this and this. You understand? Yes. So the species will be the independent variable, and then this bite force is the most correct. But I hope they also credit for those people who wrote the height of the head. Yes. Because if you are to plot a graph, you'll find out that this is A, this is B, this is C, and then these two will have two graphs in that case, whereby this is the independent. And someone talking about full marks by Darwin, that great man. Yes, state two factors what were kept constant. Factors which were kept constant here, they talked about, there are a lot, so you have to talk about any factor, but here they talked about that each species were kept in a cage with environmental conditions similar to that of the habitat, meaning that you kept maybe same temperature, same humidity, same light, same, talk about environmental factors which you could keep constant, or any other factor which you read there and then you find out that it was kept constant. So they're saying that apart from the sample size, state one way in which the reliability of the experiment was ensured. So when you come back here, apart from the sample size here, what did they do? Did they repeat the experiment? Yes, they say that the height, okay, was measured for each lizard and the average. So they calculate the average height of each lizard. You see, this is also increasing reliability. Yes. Then they are saying that school name the type of variation which is displayed in this characteristic. I told you that whatever you are born with, and it changes since you are born up to where you are now. How big was your head before when you are born, and how big is it now? How big is your size, and how big was it when you are born? So things which change like that, then it is continuous variation, because it's all individuals, they have the same. But if it does not change, like blood group doesn't change, so it means that it's discontinuous. So in this case, name the type of variation, because the size keep on changing, therefore we will say that it is continuous variation. Describe the relationship between height and the head of the lizard in terms of the bite force. As the height of the head increases, the bite force also increases. You see, 10 you see, so the bite force also increases. Which species A, B, C would be expected to be feeding mainly on rough fibrous plant? So we are looking at the one which has the force which is strong enough. So we have this, so its answer is C. Why? Give a reason for your answer. Because it has a strong bite force, so that it can chew these tough fibrous plants. Yes, so it has a strong bite force. Yes. Then they are saying that explain, so we are done with that. Which species A, B, C would be the most suitable to live in narrow areas of the rock? The one which has the small head, because it can go through the narrow or the crevices. So basically it is species A. They didn't ask you the reason, but if they had asked you the reason, then you would have said that because it has a small head. Yes, it has a small head. So that is the correct answer. Another question is 3.4. 3.4. Guys, are you getting it? If you have any challenge, let me know. Maybe you are not so sure about the answer I talked about. Yes. Let me know. Yes. Let's go to the next question, and then we try to do the last one. Okay. Which species in the diagram? What is this diagram? Is a phylogenetic tree. Yes. Which species in the diagram above existed or survived for the longest period of time? You look at how long the length of the bar. Check. It's this one. So the answer is Homo erectus. Then we come to calculate the period of million years Australopithecus afarensis and Australopithecus africanus coexisted. So here, remember I told you this, and I gave you those questions that you will never be asked a question and it will never be fail to answer it. Remember when I was going through this, and I told you here period of existence, you see, competed for resources. So that's the same question which they are asking you here, that which give two pair of species which competed for resources. So now what they did here is they gave you that. Now they're asking you here, you see, they coexisted. In other words, they competed for resources. But how long did they do that? They did that if this Australopithecus africanus is here, and then this is afarensis. Did I say afarensis? Wait. Afarensis and africanus, yes. So this is the time when they competed for resources. So basically that's the time. So I have to calculate here. So it is 2, 3, 4, 2, 1, 2, 3, 4, 5, 6, 7. So it's 2.7. Remember, we count from this side because the 0 is this side. And then this one is 3, 1, 2. So it's 3.2 minus 7, 2.7. And then the answer you get here is million years. Not million years ago, because here they are talking about the range. It's million years. So guys, I think when you subtract here, what is it? What is 2? Yeah, make sure that you subtract correctly, né? But it must have that word million years, because in most cases we mark the what? The units. Name the species that was also known as handyman. Homo, I told you this. Homo habilis. Remember when I was going through many schools trying to explain these skulls and stuff. I hope you guys you got it right, né? Got it right. State 2 uses of tools and developed state 2 uses of tools that was developed in 2.6 million years ago. 2.6 million years ago. 2.6. So it's 2, 1, 1, 2, 3, 4, 5, 6. So something which, yeah, it's this one. Yeah, the other one tools. Yes. So what is it? You just copy from here, because they said this for so there is no cramming there. Then so we got the answer there. Identify 2 species that use the most complex tool. 2 species which use the most complex tool. The most complex tool is this one. So if you look at it here, it is the Homo sapiens and the Homo neanderthalensis. Homo sapien and Homo neanderthalensis, or Neanderthals. Sometimes you call them Neanderthals, but Homo neanderthalensis. Yes. Is there anything I've skipped? Yes. Okay. Let's continue. Name 2 species. So we have done that. Explaining how the change in the brain size over time relates to developing tools. I explained this also, that increase in the brain size increases the number of cells. Yes, so this number of cells, if they are increased, then automatically it means more intelligence. And these sophisticated tools require thought. They require what? Thought. Therefore they will require more intelligence. So basically that's what they are looking for is here. We talked about it here. Cultural evidence. Using tools in cutting or improving in nutrition. Developing of tools become more sophisticated as hominids evolved. Tools require planning and thought. You see? Yes, this is what they are looking at. Tools require planning and thoughts. This is what they are looking at. Development of tools become more sophisticated as the hominids, yes, as the hominids develop. You understand? Yes. Or as the brain size changes, yes. And then they become more sophisticated, and these tools, they require planning and thought. Therefore, you have to increase the brain so that you can increase the intelligence, so that you can plan and you can also think about what you are planning. So basically that's what they are looking at there. Then we go to the next question. They're saying that name 2 species used to, okay, explain the size. So we are done with that. Let's go to the last question. Yes, the last question. Someone is saying that what if you calculate it differently but you got the answer? It's cool. It's cool. Maybe they will give you a mark. How many marks are there? Which one is it? I don't know. But if you got the answer correct, maybe because here they're saying show your answer, you're working, maybe you'll just only get one mark for the correct answer. Yes, and then you lose the 1 mark for the Let's go to the next question. 3.5. State the genus of Adipithecus, Australopithecus sediba. Australopithecus. It's this one. You have to write it in full. Australopithecus, because they're looking for the genus, the first name. Yes. Describe. It must start with a capital A. Describe the shape of the spine. We talked about this. Is S shape. They're saying describe. Just is S shape. That's all. S shape. Then they are saying that state what is Hosi Dube. Thank you very much. You are also the best because you kept yourself on the channel, and we pray that you get that distinction you are crying for, and I know you will get it. You will get it. There is one person I want to thank. Has been a member since the beginning. Oh, I've forgotten the name. Maybe I'll thank her in the next video. Yes. Yes. Okay, state what is meant by the transitional species. We also talked about this. Transitional species is a species which has both characteristics of the present species and the most recent common ancestor and the most recent species. You understand? And the most recent species. Don't say and the ancestor. Ancestor where? And the most recent what? Species or before it. This is what I'm trying to say. When I was yes, here is transitional species. This is a transitional species. It is having characteristics of this and the recent ancestor or the most recent what? Ancestor. Yes, so that is a transitional species. That's how you're supposed to connect it both in front and backward. So you're supposed to explain that in 2 versions. That's why it is 2 marks to talk about in front and 2 marks. Yeah, but in most cases here we use the all or nothing law. You either get it all or you lose it all. What if you misspell the Australopithecus? Yeah, you guys, spelling. Maybe they'll correct you if it doesn't mean something else. But if it means something else, they won't correct you, ne? So what is this? And then what is this? Do they mean the same? They have the same letter, but differently. So it depends on how you have spelled it, ne? Yes. And they're saying that give the letter of the pelvis that would be represented by, okay, give the letter. Remember, I said that Australopithecus sediba is a transitional fossil. Remember, I talked about it and I emphasized it here. Check. Australopithecus sediba is a transitional species of fossil. You see the distinction material? Yes, those people who used it correctly, I hope you're gonna get a distinction, because there is no way you're gonna fail to get it. No question can be failed to be answered by that. Okay, they're saying that give the letter of the pelvis that would be represented by Australopithecus sediba. In this case, you look at this is more wide, but it's not that wide. This is more narrow, but it's not that narrow. So this is like 1, 2, 3. So this is in the middle. So this is Australopithecus sediba. Therefore, the answer becomes A. Explain your answer, because now you have to relate these two. That Australopithecus sediba, yes, this A, the pelvis is not as wide as C and is not as narrow as B. Is not as wide, yes, meaning that is wider than this, but still is not as narrow as this. This one is much more narrower, and not wider compared to this. So it's not as wide as this, but it's not as narrow. So sorry, it's not as it's not as narrow as this, ne? Yes. Or how many marks are there? 2. Yes. So basically you would have said that it's not as wide as C and is not as narrow as B. That's the first mark. If you also wrote like this, you will get a mark. This is not as narrow as this. No, not narrow. This is not narrow. Is not as long. This skull, this pelvis is not as long as B and is not as short as this. You understand? This one is shorter compared to this, but this one is longer compared to this, which means that this is in between these two. If you look at how long it is, how long it is, how wide it is, how wide it is. You either use wide to explain, you have to compare this with this and this, or you have to use how narrow it is. So not narrow, how short it is, short and wider, isn't it? So it must be not as wide as this and not as narrow as this. Or you say not as short as this and not as long as this. This is not as short as this and this is not as narrow as this. So basically, either way or whatever you have written, then you will get those 2 marks. We go to the last question, the last bullet in the gun. The last bullet in the gun to see who got 150 out of 150. Explain the significance of change in the prognathism from Australopithecus sediba to Homo sapiens. Prognathism meaning that protruding jaws. But there's something which you need to know. When there is protruding jaws, check if this from here to here, check if this protruding jaws, basically the prognathism is reducing. Yes, because we say that this is prognathus, it's not prognathus, so it's reducing. So they are not saying increase or decrease, they're saying explain the significance of the change in the prognathism from Australopithecus sediba to Homo sapiens. So the prognathism here is reducing. So when you become more straight, flat, it means that it creates more space for the cranium to accommodate more brain. But here, because here it is not, because he has to have too many muscles so that this is being supported. Therefore, the first answer is to create or to have more brain, which indicates brain cells, and then you link them to the intelligence. Also, when it is more prognathous like this, the position also keep on changing, the position of foramen magnum. You see, if it is straight, if it is flat straight, then the position of foramen magnum indicates like this. And if it is more protruding, then automatically this position is going to have to go a little bit back so that it brings about the shape. You see, it brings about the what? The shape. But here the shape is straight. Therefore, it also allows the shift of the foramen magnum so that it can bring about bipedalism. Bipedalism, which means that you're walking on 2 hind limbs, and then this will create more faster. The organism can move more faster. So the significance here, you have to talk about the cranium becoming more bigger, creates more space for the cranium to be bigger, meaning that more brain cells, so the intelligence is linked here, while here is small. And then number two here is you talk about you can talk about it makes the position of foramen magnum to move a little bit forward, move forward, so that it can allow the organism to walk on two hind limbs, or to allow bipedalism to take place, so that now the organism can walk faster. So basically it brings about changing from the slope face to a flat face. Yeah, so that's what you are supposed to explain there, guys. I hope you got. So guys, sir, what about their deity? You're talking about, how about the deity? Yes, there are some things which I've not talked about, but you look at when you are changing from prognathism, meaning that even the jaw, the shape of the jaw will change. You understand? And if the shape of the jaw changes, because it is like this, let me show you in the book, the distinction material. You see here, ne? So the more it's checked, ne? So even this is gonna change. You see how it is slanting. When this thing is out, then automatically this one, you see. So the position is cool. So the position, we have seen it, because now it's not protruding out. So it means that the position is cool. You see now, when it is straight, it creates more space for the skull. You see? It creates more space for the skull to accommodate more brain. But here, because it is a little bit flat, so it means that the skull, the cranium becomes smaller. You understand? Yes. And then also, prognathism is basically you change. Remember, it's the jaw. This jaw, which could be also from these big teeth. You can talk about the teeth becoming more smaller. So it means that you are shifting away from the rough, tough food to rough, raw food to one which is more cooked. So that, yeah, so you have to link. Your answer was supposed to be linked. You know, I told you yesterday that once it comes to this describe thing you don't know, but you think about something which is correct. Describe thing, you write everything, and I caught it that write all your rubbish. I call it all your rubbish. But in other words, I mean that write everything you think it is correct. When you are marking, we will look for what is correct in these things whereby they tell you that describe what. Yes. So you guys, thank you very much for attending our class. You have been good people. You have been supportive. And then I wish you the best, you know? I wish you the best in your life. If you have sisters, brothers, and stuff, please refer them to this channel. If you have not subscribed, subscribe, like the video, so that it can move, and then others can also benefit. Yes, benefit. They can judge themselves, so that they go for these holidays when they are happy. If you have not made it, just this is a simple advice. If you have not made it, guys, listen carefully. Not the best in class will be the best in the world. Is it clear? So it must not make you to be too much stressed. No, I don't think, I'm not insulting, but I'm just trying to show you this. Ne? I don't think Mr. Cyril Ramaphosa, our president, ne, was the best in class, but he's the president. So that's what I'm telling you, that not the best in class is the best in the world. You understand? So that must not influence you. You understand? Keep focused. I don't think Mr. Let me say Elon Musk, whatever, yes, was the best in class. I don't think so. You understand? So not the best, I repeat, not the best in class is the best in the world. Those are two different things. I repeat, those are two different things. However much we want us to be best, but please don't lose your hope. Yes, you should not lose your hope. Is it clear? Lastly, don't treat others bad because you are the best in class. Because you don't know that this person you don't treat right might be your boss. It might be your boss tomorrow when things have not gone the way you expected them. Thank you very much. We will see you again, maybe when you're talking about the university content. May God bless you.